Hello everyone, welcome to this webinar, which is co-hosted by Menarini Silicon Biosystems and the European Association for Cancer Research. I'm Claire Sanders, the EACR organizer for this webinar. I will give a brief introduction before handing over to our session chair. This webinar should last approximately 1 hour. We'll have a short introduction from myself and Menarini's chair, Nicolo, before the main talks from Bernhard, Polzer and Cäcilia Köstler, which will be around 20 minutes each, followed by 20 minutes for questions. You can use the Q&A box on your screen to type a question at any time, but please note that all questions will be saved until the end of both talks. Finally, from me, I would like to tell you a bit about the EACR. I know many of you are members already, so welcome back to all of you. If you've not heard of the EACR before and you work in cancer research, I invite you to join our community. You can find out more by scanning the QR code on screen. We have 12,000 members worldwide across more than 100 countries. Membership is open to anyone involved in cancer research, wherever they work, in any field, at any level. I wish I could get an EACR member to come on screen right now and tell you about the EACR, because I think our members can best describe what the EACR means to them. Over 99% of our members say they would recommend EACR membership to other researchers. We exist to support cancer researchers in their work and careers. That includes organising great conferences, providing access to different funding programmes, educational and career webinars, and tools to help network and collaborate. On top of all the tangible benefits for many EACR members, the best thing about membership is the sense of being part of an international community with the same shared goal. Menarini Silicon Biosystems is one of 17 EACR industry partners who work with us and support the work of the EACR. One of the ways we collaborate is to organise webinars like this one. I hope you enjoyed the session and we look forward to seeing you at another EACR event soon. I'd like now to hand over to Nicolo Manaresi. Nicolo got his degree and PhD in electrical engineering and computer sciences from the University of Bologna, Italy in 1993 and 1999 respectively. He was research assistant at the Swiss Federal Institute of Technology in Zurich and co-founded Silicon Biosystems in 1999. He was president and CEO through the early stages of the company and now serves as CTO. He has co-authored more than 50 scientific papers for which he has received an award. And he is also a co-inventor behind more than 30 international patents. So over to you, Nicolo.
Thank you, Claire. It's my pleasure to be here. And I'm really excited to listen to the presentation of today's speakers. At Menarini Silicon Biosystems, we are striving to bring to the market technologies which can make a difference in the clinic. So eventually our technologies aim is to help patients, and we will learn a lot about this from today's speaker. The first is Bernhard Polzer, and Bernhard has studied medicine in Munich and worked as a scientist at the Institute of Immunology at the University of Munich and the Institute of Pathology, University of Regensburg. Together with Professor Christoph Klein, he has established the first certified diagnostic lab for single-cell analysis. And he's currently deputy head of the division of personalized tumor therapy at the Fraunhofer Institute for Toxicology and Experimental Medicine, always in Regensburg. So within this role, he's heading the group of molecular and cellular diagnostics, which focuses on the development of single-cell technologies in liquid biopsies and the clinical application of diagnostic assays for systemic cancer. So Bernhard, I hand over to you the stage. Please go ahead.
So thank you very much, Nicolo, for introduction. And thanks, Claire and the EACR team for giving us a chance to present here at Menarini Silicon Biosystems for course 2. I try to share my screen next, just a second. Claire, I can't choose my screen. I'm sorry.
Sorry about that. Try now.
Okay, I started again. Yeah. I tried to do it with whole. Let's try it like this. This is working. Okay, I hope you all see the presentation mode now. So in principle, first of all, thanks for the introduction, as I said. And I want to mention that the first part, so my part, is actually pretty boring. So I will not talk about CSF yet. This is the task of Cäcilia, and she will present all the exciting data that we generated on this topic. But I will give a short introduction. So what we are doing and why the technologies are important and why you have to take a good look and always think about how to use available technologies for certain applications. So in principle, I don't have to talk too much about the concept of liquid biopsy, because I guess all the people, or nearly all the people here, know about this. So the concept is that we try to get molecular information on systemic cancer with a simple blood test. So no tissue biopsies, just looking at the blood contents. And many people know about circulating tumor DNA. So this is basically one of the main focuses today, but there are other options: circulating tumor cells, microRNAs and other cell-free RNAs, and also extracellular vesicles. Our talk today is mainly about cells. So in this case, from blood circulating tumor cells. And if you are working with circulating tumor cells, you have to deal with a problem, because blood, there are billions of cells in a very small volume. And if you want to find the needle in a haystack, so a very small number of circulating tumor cells in these billions of blood cells, you need technologies to enrich and detect those cells. One example that I guess most of the audience knows is the cell search system. In the cell search system, basically, you plug in your blood sample by some preparation. The cells are enriched for EpCAM, an epithelial marker, and then stained for cytokeratin, DAPI, and CD45. And in principle, what you get is those galleries with images. And here you see one example of a circulating tumor cells. It shows the cytokeratin signal in green, the DAPI signal in purple. And also you take a look at cell morphology and size. So is this roughly depicting a cell? And it should be CD45 negative as a marker for blood cells. So this is how cell search works. And this is implemented pretty good in the community. And there are many data about this. This is just one example on breast cancer, where it could be shown that in many, many patients, if you detect 5 or more of those rare CTCs in 7.5 ml of blood, that this makes an impact for the prognosis of the patients. And this led in the end to the FDA approval of the system for breast, prostate, and colorectal cancer. However, I'm not sure if this is the end, because what is the therapeutic consequence by just looking at prognosis? And now we have to go one step further. And this is basically, when I go back to this slide, it's not only enough to detect cells, you somehow have to characterize them. You have to define, are there mutations, are there proteins of interest on the cells that can be attacked or approached by therapies? And there, another technology comes into play. You have to isolate the single cells that you have enriched and detected. One example, again, the DEP array from Menarini Silicon Biosystems. And then you have to do molecular, or in some cases, even functional analysis with the cells to get a readout, to get some idea what can the clinician do for the individual patient. And on this molecular analysis, I want to spend the next part of my talk. So in principle, you always have to consider, if you're dealing with one single cell, this is pretty low amount of material. One cell has roughly 6 to 7 picogram of diploid genomic DNA. So if you want to analyze this comprehensively, you have to do some sort of amplification before you go to downstream applications for sequencing, for example. And this is done usually by whole genome amplifications. And if you want to use this in a clinical setting, you should have some prerequisites of these methods. So it should be robust and should be rather homogeneously amplify the whole genome of a single cell. You need sufficient sample volume for test repetition. If you're doing research, also archival storage for later repetition, new tests that may be interesting in the future. And very important for the tech development, it should be compatible with different downstream applications. And because, I mean, the field is approaching very fast, and basically every 5 years, you have to kind of change your whole readout repertoire. So this is why I want to come to this table. This is some years old, so for sure not all the available whole genome amplification technologies are in this list, but the principle that I will show you now is still holding true. So if you look at the first line, you see that in principle, there's two mechanisms to amplify the DNA for single cell. It's either a PCR-based method or it's MDA, multiple displacement amplification. And if you take a closer look at this table, at this line, you see these are results from publications where people isolated a single cell, did the whole genome amplification, and then a whole genome sequencing. And if you look at this line and look at the coverage, which is deviating from 36% for Picoplex to 93% for the Malbach method, of course, the higher the number gets, the better. However, already here, I want to make a point. So genome coverage is always only looking at haploid genome, so basically not on different alleles, which is important for cancer. And the second thing that you should know is that this is basically always in an experimental setting with a perfect cell. So back then in 2013 or 2012, Malbach was introduced to the field. There was two big publications. I think it was Nature and Cell, or Science and Nature side by side. And everyone was talking about Malbach. And this method basically was used a lot in this time. And this was then the first paper that really dealt with clinical samples. And this is actually a very important paper. What they did is they took one patient with lung cancer, and they had a sample of the primary tumor, and they had a sample of the metastasis. And back then, it was not clear yet if CTCs that you detect in the bloodstream reflect the metastasis or the primary tumor that was already removed by surgery. So is it a measurement for the systemic load, or is it only something for the local disease? And what you can see here in the different colors, so gray means different mutations are analyzed and listed. Gray means it's wild type. Blue means you find a heterozygous mutation. And this dark blue, which is actually pretty rare, means you have a homozygous mutation. So the wild type allele was lost in the metastasis in this case. So the next step that the authors did is they found many CTCs in this patient. So this was one of those rare patients with hundreds to thousands of CTCs. And they isolated many of them and selected the 8 best, depending on their quality criteria. And if I now go to the next slide, you will see that the pattern that you see in those CTCs in general more closely reflects the metastasis than the primary tumor. However, there are mutations in this area here that are not found in the metastasis, but in most of the CTCs. So maybe they reflect a subclone. And there are also mutations like above here, where you don't find the mutated allele in the CTCs, but you have it in the solid metastasis. However, if you take a closer look, now another color appears, white. White is not defined in the legend. And there's much more dark gray and much more dark blue and much more gray. So white means I do not detect the sequence. So how is so much white working with 93% of genome coverage? And there I want to repeat, this paper was about fresh cells from the cell culture. This here are patient cells. And again, you see the many dark blue spots, you see the many gray spots. You can't really say, for example, if in CTC5 here, those genes are really not mutated, or if just the mutated allele was lost, or here, if the wild type allele was lost. And there was another paper by a Dutch group who basically took the data from the PNAS paper and used the same algorithms that the group that invented Malbach used for their Nature publication. And you see that the data here is not reflecting 93% of genome coverage, but only 33%. So this is a very important point. Cells from the patient have to be shipped to a center. They are fixed usually. And this all has a great impact on the WGA technology that is used later on. And then again, what I highlighted also, look at the colors, there's high random dropout. This is problematic, especially for cancer, because you always usually have a wild type and a mutated allele, and you want to have all the information. Back to this table, I want to highlight those two actually pretty boring on the first look lines. And here you find highlighted in green, in red, that all of the different methods have a random step, all of them except ampli1 WGA. And why is this important? Because if you know and have defined what you can do, and this is how the method is working, basically the cell is digested, then we digest the double-stranded DNA with an restriction enzyme, we anneal a universal adapter, and then we amplify this. So by this method, you basically know how your library in the end looks. And this is very important because when you know this, you can design primers. This year, it's already some years old, and the younger ones of you can't imagine this. Back then, 2014, not so long ago, Sanger sequencing was pretty much done everywhere. NGS methods were not so common, and this is Sanger sequencing. What we did is we isolated a bulk DNA from a cell line, MCF7 is a breast cancer cell line, and looked for an oncogenic mutation in PI3 kinase exon 9. And here you can see this is the location where you find the mutation. So one allele is a T, the other one a C. It's a 50-50 mutation in the bulk DNA. We then isolated 20 cells amplified by 12 cells amplified by ampli1, and then you can see similar to the bulk DNA, in every single cell we roughly find 50% of the sequences are mutated, 50% are wild type. So as well the allelic dropout, also the coverage, the assay principle works in every single cell. Why is this important? If you now transfer this and go one step further to whole exome sequencing, what there is done, you have the whole genome, you shear the DNA to get smaller fragments, and then usually capture probes are used to basically catch the exomic sequences, use them for sequencing, and get rid of all the interloping DNA to save sequencing power. If you have a random method, those fragments are different in every single cell. However, with ampli1, you really know where are the cutoffs. And now, of course, you also know that some sequences should work and others will never work. While here it's basically random. There might be the perfect cell, but there might also be cells where you don't get much information. And this is again the PI3 kinase gain. The exons are depicted here with the dark blue boxes, and you can see in 3 biological replicates, 2 technical replicates for each biological replicate, you see that basically in every single cell you get a very similar picture. You get the sequencing from the exome that are covered by MSE1, but you also know by in silico analysis that there are some exons that you won't get. So here you will not get any result with the method. Here you will get the result. This is pretty important if you come to diagnostic approaches. So of course, this is not everything. So in our lab, we established nowadays many different technologies on the DNA and also on the RNA. We even can do both out of the single cell. I don't want to show off here, but I want to highlight one thing, and this is the second point that is important when you're dealing with clinical samples. You really need good QC if you want to deal with patient samples, because not every cell is the same. And also there, it's very good if you know what you have in your sample, then it's easier to devise a QC. That this works, you can see here, this is 120 cells from melanoma patients that passed our QC criteria, and we did whole exome sequencing. And in principle, all of the cells except this one here are in the corridor of 75% to nearly 80% whole genome coverage. So this is the advantage of a non-random whole genome amplification method. Now coming back to this scheme, of course, blood is a little bit problematic. So you have rare target cells within a very high cellular background. You need highly specific markers to detect them. And then isolation is actually pretty straightforward if 1 and 2 really work. However, this last point is very important, that molecular analysis, again, depends on the quality of the DNA and therefore a little bit on the cell viability. And there we have a problem with CTCs because they are frequently apoptotic. In our first paper from 2014, we could basically show that also already morphologically, you can define different categories of CTCs. And even if the cell looks pretty good, you see here with a very simple PCR assay that many cells are total dropouts, 20%. And only 33% of the CTCs that look very good give you really high quality, highest quality results. Nevertheless, we applied this in a large study, and this was basically samples from the text studies, German multi-center trials, different ones, really with treatment based. So we got cell search samples, we isolated the cells, we did whole genome amplification of the cells with a isolated with a deparate, did whole genome amplification, then did the QC, which is basically now the QC kits from silicon to determine the genome integrity index. And then we did low-pass CNV profiling. And in this case, we really, because with CTCs is not so easy, took all the cells with GAI 1 to 4. So whenever we could identify human DNA in a sample, we went on to sequencing. And you see here from the numbers that only 65% gave us something. So you have a dropout of roughly 35% of the cells that we isolated. The results, however, of those tests are pretty good. So first of all, also this is important. Depending on the GAI, you can already get a first glimpse that the higher the GAI, the more fragments you detect with this very simple assay, the higher the chances that your low-pass sequencing, whole genome sequencing will work. So GAI gives you worse results, GAI4 gives you the best results, and 80% of those cells work. This is results that you can get. Of course, now we are starting to look into the data. You can look for intra-patient heterogeneity. So are all the single cells of this patient the same, or are maybe subclones available, like here, what you can see with this loss on chromosome 5, maybe shortly. So this is a CNV profile from chromosomes 1 to X. And you always see the median is the gray. This is basically baseline, it's a diploid genome. And when you have blue, you see losses. When you have red, you see gains of respective chromosomal regions. So here, basically, you see that they are very clonal, but there are subclones indicated, for example, by this loss on chromosome 5 in this cell in the lower part of the picture. Then you can look at loss of heterozygosity. This is also interesting, depicted by the color coding here. So here you have an example of chromosome 7, where we have a gain and a loss. And you see that here, this is only one allele. And while here you don't see the loss, still the wild type allele or one of the alleles is lost. So you have two copies, but of the same maternal or paternal chromosome 7. And then, of course, you can go a little bit into looking at genes by identifying high-level amplifications and looking which corresponding genes lie there. And in some cases, like cyclin D1 or HER2, it's pretty straightforward. If you have this amplification in this very small region, then it's very likely that HER2 is amplified in these cells. So coming to the end of my talk, I want to repeat this slide, because this basically is now introducing what Cäcilia did in her thesis, because we were asking ourselves the question, if there's a problem with blood because you have so many background cells, could we imagine a scenario where we don't have all the problems depicted here, so basically not such a high background, and very important, viable cells that are available for analysis. And so if we could get rid of this, maybe we have a better chance to bring single cell analysis into the clinics. And this is where we started to work on the transfer of liquid biopsy concept to other organs and clinical applications. And now this will be taken over by Cäcilia in her talk. She will talk about the CSF applications that we developed in the last years. In the end, I just want to say and to repeat, if you're working with liquid biopsy, you have to be comprehensive. You have to care for many things: sample preparation, pre-analytics. You have to select the right technology for your molecular or functional analysis. And of course, you need special bioinformatics, because single cell data is not the same than bulk DNA. And in the end, I also want to thank my team at Fraunhofer ITEM, the other groups and group leaders there, our collaborators, especially Christoph Klein in the single cell diagnostic lab in the University of Regensburg, and of course the clinical partners. And I want to highlight our partners from the BZKF, the Bavarian Center for Cancer Research on Brain Cancers, and the DETECT study group, led mainly by Wolfgang Janni and Tanja Fehm from Ulm and Düsseldorf. And of course, Nicolo and the team at Menarini, and also Claire and the EACR team, and all donating patients and our funding agencies. And then, Nicolo, back to you.
Okay, thank you very much for this excellent presentation, very clear introduction about the challenges of single cell analysis and how important it is to care about the quality of the reagents and the methods that you use in order to transfer them into the clinical applications. So now I'm pleased to introduce Cäcilia Köstler, which has studied in Würzburg, Germany, then at Max Planck Institute of Biochemistry in Munich in the field of protein biochemistry, with broad expertise with biomaterials, bone grafting at the University of Beirut. And finally, she landed in 2013 at the Fraunhofer Institute for Toxicology and Experimental Medicine in Regensburg, always in the group of Professor Klein, and where she is a specialist for single-cell enrichment, isolation, and molecular downstream analysis. She recently won the Poster Prize in 2019 in Bergamo for the EACR Liquid Biopsy Symposium, and she's leading project at the University of Regensburg while she's finishing her PhD about cerebrospinal fluid liquid biopsy, which is the topic she is presenting today. She's also very busy as a mother of 3 children and a woman in charge for gender equal opportunities for her working environment. So, Cäcilia, I hand over to you.
So thanks, Nicolo, for your kind introduction and for giving me the opportunity to present my data in this format. And welcome everybody from my side to my part of this webinar, where I'm going to present my PhD thesis, where I extended the liquid biopsy concept from blood to CSF. So, leptomeningeal metastasis is the spread of disseminated cancer cells to the subarachnoidic space and the meninges. So 10% of metastasized tumor patients are affected, and the most frequent primaries are breast, lung, and melanoma. The overall survival of treated patients is up to 5 months. And if patients apply to the clinic, they come with symptoms like headache, mental changes, or motoric dysfunction. And if they apply to the clinic, the first evaluation is to clarify the symptomatology, and the standard of care is to analyze the cerebrospinal fluid. Also, images are performed from the brain and the spine, and biopsies are very rarely performed. There are different therapy possibilities in combination or singularly, the systemic therapy, chemotherapy, or radiotherapy. And this is dependent on the molecular data, mostly from the primary tumor. But since the diagnosis cannot always be made on the basis of molecular data of the primary tumor, the imaging is not always clear and biopsies are very rarely performed. It is important to close this gap and move towards sensitivity, quantitative determination of cells in the CSF, and their molecular analysis. So therefore, we started a local study with patients with suspected leptomeningeal metastasis and investigated first what is the comparison between the results of the imaging data and the neuropathological cytology as the standard of care. And what you see is in the direct comparison is that we have 2 methods which show a comparable sensitivity in order to detect positive or negative samples. But if we look a bit more detailed, we also detect in 33% of the samples discordant results. And now we come to the question, how can liquid biopsy improve this clinical situation? So we started a proof of concept study in order to compare our data to the cytology data, and therefore we analyzed cerebrospinal fluid. For the cell-free DNA part, we used the supernatant to isolate cell-free DNA and quantify. And with an adapted in-house library protocol adapted from our single-cell workflow, we amplified the cell-free DNA, and then to start a copy number variation analysis by the AMPLI-1 low-pass library. For our DCC enrichment, we used the cell fraction of the CSF and enriched the EBCAM positive and cytokeratin positive cells by the cell search system. This is an FDA-approved system for blood, and we adapted, as known already in literature since years, this to the CSF. We tried to quantify the DCCs and then isolate and amplify them by the AMPLI-1 whole genome amplification for a later copy number variation analysis with this AMPLI-1 low-pass workflow. So our sample collective included 28 patients with the suspicion of leptomeningeal metastasis, but different primary tumors and also cancer of unknown primary. And we had the chance to compare 28 samples with the cytology approach and our liquid biopsy approach. For 21 of these patients, we could get imaging data, and the cell-free DNA analysis was performed on 20 samples from this collective. So can we detect CSF tumor cells by the cell search system workflow? So in 40% of our samples, we could find positive cytokeratin cells. And we could show that with our workflow, we have a qualitative and quantitative analysis option. Our workflow is high sensitive for samples, also with a low cytokeratin positive cell numbers, lower than 5 DCCs. But could we also generate with our workflow data comparable to the neuropathological cytology? And in this direct comparison, we can see that in 86% of our results, the results were concordant between the cytology and the cell search workflow. So we have 4 different results, but 3 of them were positive with the cell search workflow, 1 negative in the cytology, and 1 was negative for the cell search workflow, while positive in the neuropathology. But this is a cancer of unknown primary, and which might be that the markers included in the CTC kit were not specific enough for this cancer type. But in the end, we could say that our CSF cell search workflow is at least as sensitive as the neuropathological cytology. But can we now confirm the malignancy of the single tumor cells on a molecular level? So therefore, we isolated 144 cytokeratin positive single cells and 14 CD45 positive single cells from our samples. And we measured the quality of our DNA from the cells with the genomic integrity index Bernhard mentioned before already. It's a PCR-based multiplex assay where we define the quality via the genomic integrity index. And what we saw is that in 93% of our CSF cytokeratin positive samples, we have a very high DNA quality. And compared to the publication from Bernhard from 2014, it's much higher than the single cells isolated from blood, also with the same workflow. So with these high quality cells, we could go on for all samples with the CNV analysis. But how does this look then on the molecular level? We could, for 97% of the cytokeratin positive cells, display the evidence of tumor origin. As you see here in the upper part, it's an aberrant CNV profile of a cytokeratin positive cell. We isolated from the CSF of a breast cancer patient. Compared here below, you see a CNV profile of a CD45 positive cell with a balanced profile, and this was also isolated from the same sample of this breast cancer patient. And for all of our CD45 positive cells, we got a balanced profile. So to sum the benefits up of our cell search workflow, we have here a workflow which gives us equivalent results to the neuropathological cytology. It has a high sensitivity to detect also DCCs in samples with a low cell number. We could generate high quality DNA samples, and all cell search positive CSF samples contained disseminated cancer cells. Furthermore, we generated with this workflow a whole genome amplified material for further downstream analysis. And we have, due to saving the samples in the cell-safe tube, this is the preservative tube with a mild fixation included in the kit that does not harm the quality of the cells, a very time-flexible workflow where samples can be processed up to 96 hours. So, but I told you before that we have 40% of our patient samples with the suspicion of leptomeningeal metastasis, which were positive. But what about the negative samples? So can maybe cell-free DNA complement these results from the single-cell analysis? And therefore, we analyzed 20 of our samples for the cell-free DNA. It was possible to enrich cell-free DNA for all of our samples, and we could reach a sufficient concentration of cell-free DNA for all these samples in order to perform the CNV analysis of the cell-free DNA. So up to now, it looked like we found a time and cost-efficient method, which might give promising molecular data. But if we go now more in detail, we see that in 25% of our samples, we could confirm the aberrant CNV, the tumor origin by the aberrant CNV profiles. In 45% of our samples, we got a balanced profile, and we have a dropout of 30% where the cell-free DNA samples were not evaluable due to the low quality of the sample. So if we compare now the DCC data to the cell-free DNA data, we saw that with the DCC workflow, we get 100% meaningful results. We don't have any dropout, and we confirmed 100% of the positive cytology results compared to the cell-free DNA, which was only possible for 42%. So compared both methods for positive samples, we could detect 5 samples with cytokeratin positive cells by the DCC workflow and 2 samples only by the cell-free DNA analysis. And to bring this together to the data of the cytology, we can say that cell-free DNA is a cost-efficient and fast method also to generate molecular data, but we have a 30% dropout, and samples with a low cell number are balanced in the cell-free DNA analysis CNV profile. So cytology is a more reliable workflow than the cell-free DNA workflow. But as I told you before, with 2 samples, we could only detect the positive leptomeningeal metastasis samples by the cell-free DNA analysis, nor by the DCC enrichment, neither by the cytology. And that's why we think it might be ideal to combine both CSF liquid biopsy methods to first check in a quick and dirty cell-free DNA part if we can get molecular data which were sufficient, and in the meanwhile enrich the cell fraction with the cell search system, and if it's necessary to isolate and molecular describe the single cells isolated from the CSF. So because we wanted to have our workflow maybe included into the clinical daily routine, we wanted to know if we can use the cellular liquid biopsy to guide treatment decisions. And therefore, we started an ongoing study to identify brain-specific alterations by comparing different lesions and the primary tumor. Therefore, we used the Deborah FFPE sample prep workflow, where we dissociated tissue sections up to 50 micrometer from primary tumors and different metastatic lesions. And by staining for cytokeratin and vimentin, we could use the Deborah technology to define the tumor cell fraction and the stromal cell fraction, and to isolate the pure cell populations to single tubes. After the amplification, we of course performed the CNV analysis to compare the data. In total, we isolated 26 tumor cell populations and 19 stromal cell populations, and we could perform the CNV analysis on 80% of our samples. And what you can see here are two samples from one patient. The upper one is the tumor cell pool of a peritoneal metastasis from 2016, and the lower one is a CNV profile of the tumor cell pool of a bladder metastasis from 2018. And what you can see, both CNV profiles are aberrant, but they look quite similar. And if we now enlarge these molecular data and compare it to the molecular data we got from the CSF analysis, we could see that samples derived from the same patient, that it's possible to trace clonal evolution by different biomarkers. This is nicely shown in this circus plot, where on chromosome 8, 9, 11, and 18, the clonal differences can be seen between the samples derived from the central nervous system and not from the central nervous system. And that this cellular liquid biopsy might be a good possibility to predict therapy by brain-specific clones. We showed this with the first clinical sample by publicating data in 2018. There we showed a case report from a cancer of unknown primary patient. She came to the clinic with headache, nausea, and vertigo. And in the cranial MRI, no tumor was detected. But in the CSF, there were conspicuous cells, TTF1 positive and cytokeratin 18 positive, which led to the suspicion of a lung adenocarcinoma. So the CT showed an intrathoracic mass in the lung, but the biopsy was tumor cell negative and the PT scan was also negative. So the clinicians decided to analyze blood and CSF for single cell enrichment, and no CTCs in blood, but 14 DCCs in CSF could be enriched. So after isolating, they were molecularly analyzed, and some of them showed an EGFR missense mutation. And with this, the patient could be treated with afatinib. So to sum up the most important details for you, we could see that our CSF cell search liquid biopsy workflow is a qualitative and quantitative method for single cell enrichment. We could isolate high quality single cells and confirm the tumor origin of each CSF cytokeratin positive sample, and that this is a good alternative method to the conventional cytomorphology, which is still the actual gold standard in the clinic. With our cell-free DNA isolation of CSF, we could generate additional molecular data, but it is not an alternative method, neither for cytology nor for the DCC enrichment, but it can be a fast and cheap complementary method in addition to the daily routine in the clinic. And by our tissue analysis, we could get molecular confirmation from different time points of the disease, and we could also define different subclones, but the data in this part are very single cases and have to be enlarged for a better evaluation. And with this, I want to thank to my doctoral thesis supervisor, Christoph, who gave me the opportunity to work in his lab and who never stops believing in me. I want to thank also to my direct supervisor, Bernhard. He's also my group leader. He's also supporting me since years and also never stops believing in me. And thanks to all my colleagues who supported me during this work, all collaborators for their kind support in scientific discussions, and the EACR and Menarini, which gave me the opportunity to present my data today. And of course, to all patients who were willing to give their samples. Thanks for your attention, and I'm looking forward to your questions.
Thank you, Cecilia. Maybe we should now take questions. I've seen some already coming in the Q&A box.
Yeah, if I could just take a moment to give an overview of how the Q&A session will run again, in case anyone's not sure.
Yeah.
So I can see many people have already found the Q&A box at the bottom of the screen, so that's great. If you haven't, then it's available at any time. We would very much like this to be a discussion, so we encourage you to ask any questions. The EACR has a motto that there are no silly questions, everyone's learning, and you can ask anonymously if you prefer. We would like this to be interactive, so you will be given the opportunity to ask your question verbally. If Nicola reads out your name, then I will send you a prompt to turn your microphone on so you can ask your question directly to the speaker. Nicola will read out any questions that are posted anonymously. So I'll hand back over to you, Nicola, and as you said, we've got a few coming already.
Okay, so sorry, you said first the anonymous question or the...
You can read whichever one you'd like.
Okay, because the first one incoming is from Marina Puchinskaya. I'm not sure whether she wants to speak for herself or I should read.
Good evening, do you hear me?
Yes.
Thank you very much for this opportunity to ask a question, and thank you very much for wonderful data. So I've got really two questions. I'm a clinician scientist and I'm mostly interested in EMT and cancer stem cell markers, including in circulating tumor cells. So one question is, can this method of CNV analysis or whole genome analysis, can it help with detection of complex transcriptional programs like, for example, EMT program is included in CTCs? And the second question is regarding cell search methodology and regarding the use of cytokeratin and vimentin to differentiate epithelial cancer and stromal cells. There are many data in the literature that possibly cell search technology may not be useful enough and efficient enough in detecting circulating tumor cells with EMT features due to loss of cytokeratin. And so it may just not find the cells that may be really a source of metastatic spread, including in leptomeningeal metastasis. And again, vimentin can be really expressed on cell surface of epithelial cells with EMT features. There are also data in the literature. So how could you comment on that in your CSF samples? So are you sure that vimentin positive cells are stromal and are not cancer cells? Thank you very much again.
Okay, so maybe I don't know if just to recap this very long question. So the first one is for maybe for Bernhard regarding the RNA, and then for Cecilia to the CSF and the EMT question.
So it was a very complex or many questions. Thank you for them. I will try to answer them. So first of all, with the workflow that you saw today, including the cell search system, what you can do is DNA analysis, because I mean the cells are fixed, they are dead. So usually you can get high quality DNA, but the RNA is in most cases degraded, and because of the cytokeratin staining, where you perforate the membrane, basically RNA is also lost from the cells. However, of course, it's possible to do RNA sequencing also with single cells. I mean, we are doing this, but the stuff that I said about the cell viability is, of course, very important. So from the CSF, we have another study, not with metastasis, but we have a study on rare brain cancers where we do exactly that. We isolate tumor cells from rare pediatric brain cancers. We do transcriptome profiling of very rare cancers to look for new biomarkers. So this is feasible. You just have to keep in mind that, of course, for RNA, the window is much lower. So at the moment, I would do this locally. You need the sample very quick. You have to analyze the cells rather quickly, and then it works pretty good. There might be options in the future. I mean, we had the chance to test the new cell rescue tubes from Menarini Silicon Biosystems, which are an alternative cell safe. And at least in our hands with cell lines, we could show that even after 2 days, you can get meaningful RNA results out of this. I mean, this is just a proof of concept, not published yet. But at the moment, I would say for good RNA analysis, you need another approach and you need fresh cells to do this, but it is feasible. However, with RNA, of course, the noise is also bigger. So you have to be pretty sure you need good markers.
Thank you, Bernhard. Also, just one comment before ending to Cäcilia. Just in the last week at the EACR, there was, I think, an abstract comparing the cell rescue performance for RNA analysis with other commercially available tubes, which confirms that it's actually the best in class for preserving the RNA for single cell analysis. So concordant to what your comment. Cäcilia, your comments on the CSF possibility and the EMT from the cells that you collect from CSF. Is it possible to?
So I think it's as Bernhard told already, but we did not check this in this project. But what we see is that with this cell search tubes, as Bernhard already mentioned, also the cell rescue tube is very good mild fixation to not to harm the quality. But for EMT, we did not check. I cannot answer this question on this special project.
Maybe I can add, Marina, so you are maybe going back with a good feeling. So, of course, cell search itself enriches for EpCAM and stains for cytokeratin. So if your cells do not have both of these markers, you won't get a positive results. I mean, what I can tell you by our data that, of course, everything is possible. I mean, my MD thesis 20 years ago was basically on single cells in the bone marrow in prostate cancer. And I defined that by just using those two markers, EpCAM and cytokeratin, you can detect different subpopulations. And I mean, it might be different for every cancer type or every tumor. At least in prostate cancer, from our data, I would deduce that cytokeratin is more showing differentiated cells, while EpCAM only cells might be more undifferentiated, more driving the disease. And I think this really can't be answered in general. I think this depends on the tumor and it depends on the setting. We also know from our results that we also generate models. Another group in our lab generates models, and it's not really correlated to the CTC number in the cell search in some cancers. And sometimes we don't find any cytokeratin positive cells with cell search, but with other enrichment methods, we find thousands of EpCAM positive cells and we get a model. So I think this is a very tricky question, and also vimentin alone will not be the answer. So I think depending on the cancer type, depending on the maybe individual cancer patient, everything is possible.
Okay. Another question here from an anonymous attendee. This for Cäcilia. How is the cell-free DNA amplified before low-pass analysis? Are you using the whole genome amplification or a variation about that?
So it's not yet published an adapted in-house library protocol. I can not let you know until the data will be published very soon, but it's not the whole genome amplification. But I think, yeah, you will be, it will be published very soon. And
looking forward to the publication then.
It's still a hidden protocol.
And for Bernhard, what is the value of measuring loss of heterozygosity in single circulating tumor cells?
So, very interesting question. I can't really answer this. I just can give you one example. That's why I think this can be possible. I mean, for example, there's a breast cancer cell line. I don't remember. I think it's CAL51. When you do CNV analysis, you see that this is a flat profile. So it's a balanced profile. But if you're looking at loss of heterozygosity, you suddenly see that many regions or whole chromosomes, one copy was lost because you then have two copies of the one chromosome. And I think, Nicolo, this is making more a question for you because I think you're more into this topic. But I think this is invaluable information. And you also saw this in the example that I showed. in this one patient, the flat profile in the CNV profile would suggest there's nothing happening, but when looking at the LOH, you see it's only one type of the allele, and this might matter.
Yeah. Also, maybe I can add to this that loss of heterozygosity is important for predicting therapeutic efficacy in homology recombination deficiency, to assess homology recombination deficiency, which is linked to efficacy of PARP inhibitors. So as they are becoming suitable options for many type of cancer, breast, ovarian, I think this will become more and more important. And this is basically unfeasible with ctDNA due to the dilution of wild type DNA. When you have a single cell, you have a pure cell, and from the low pass, having the possibility to detect also this kind of information may prove very valuable, I guess. So another question is for Cäcilia, what is the volume of a CSF that is processed for the cell search workflow? And if there are other preparatory steps before the cell search analysis that you may disclose?
Thanks for this question. So we performed the CSF analysis with the cell search system in volumes between 900 microliter to 7 milliliter around. But what we did is, because the cell search system is normally working with blood. Of course, there are a lot of publications on how to use the cell search system for the CSF analysis, or not for blood analysis. But what we did, we shared somehow by centrifuging the sample to the supernatant and the cell fraction, and the supernatant for the cell-free DNA and the cell fraction. We then took the pellet around this 300 microliter out of the volume of the CSF and to fill up with the buffer, and then let it run in the cell search system. But the volumes were up to from 900 microliter up to 7.5 milliliter. It depends on the sample. When the CSF sample comes, it's not always a standard volume what you get. It depends on what we get.
And another question from Pia Zeiner. We want to...
Hello, do you hear me?
Yes.
Thanks for the interesting talk. Yeah, I was just wondering how you did extract the cell-free DNA, because you had amazing yields, given that the input volume was, I think, on the slide up to only 2 milliliter. So did you use a kit, or how did you do it, and how did you do quality control?
As I showed in this slide, we were using the QIAGEN circulating nucleic acid kit for enrichment of the cell-free DNA, and then we performed the qubit assay in order to detect. But then this high yield we reach, of course, by our not published yet adapted in-house library protocol. And that's why I cannot answer this question up to now. But maybe Bernhard, you want to say something about this?
So yeah, maybe I add to the two questions that came before. We cannot disclose this yet, but I mean, in principle, what we also saw, I mean, this comes to your question. We started with children, and in children, I have to say the tumor load in the CSF cells and cell-free DNA is a little bit hot. You get lower volume, but the load is higher, so it's more concentrated in small children. However, in the adults, we had the problem, and this is why we actually invented the method, that very frequently could not measure any cell-free DNA. And then Giancarlo in our lab and Cäcilia helped him develop a method where we, in principle, by introducing different steps, bring the cell-free DNA in the single-cell workflow. And then you get those yields. And as you can see, the problem that we basically get out of this is that we get DNA in all the samples, but in some 30%, basically you start with just contaminants, and this is why you don't get any results. This is just very noisy profiles. So by introducing this amplification error, we make many more samples available, but the quality control is a problem because you cannot really so easily determine those dropouts. I hope this answers your question.
Yes, thanks so much. Curious for your publication.
Okay, so another question is whether you have been also looking at extracellular vesicles from the CSF.
Do you want to answer, Cäcilia?
So up to now, this is an ongoing study, and we also enrich or enlarge our portfolio. And we tried to start it to analyze also extracellular vessels, but we don't have any data so much ready to present.
And maybe also to add there, this comes back to this very low volume. So our priorities were, you see this in the talk by Cäcilia, our priorities are the cells, then came the cell-free DNA, and basically the numbers for extracellular vesicles were even lower because you need lots of supernatant that you can basically get vesicles and cell-free DNA out of there. So it's a limited resource. So we did it. We have preliminary data. There's also interesting data in this very few cases, but yeah, this is not so far that we can really publish something.
Okay, so another question is whether you tried to isolate cell-free and or cell-free DNA in the same cerebrospinal fluid sample that was used for cytology analysis. So were they matched? So Sandra Monguera, sorry, do you want to elaborate on your question? I was reading. Can we give the microphone to Sandra, or?
It looks like they might not have a microphone, so you
okay.
can carry on.
Okay, so thanks, Sandra, for this question, but this is what we basically showed. So we took the CSF sample, we split it. One half was analyzed by the neuropathology cytological workflow, and the other half went to our lab. And that's what we saw, is that in 86% we had these concordant results between the cytology and the cell search workflow. And additionally, there were 4 different examples. 3 were positive for the cell search workflow, and 1 was positive only for the cytology workflow. And this was this cancer of unknown primary I showed in one of these slides. Does this answer your question? I hope that we splitted the sample and it was analyzed in parallel.
And to add, cell-free DNA was also of the same sample. And important to add this again, so the 3 samples that were positive in cell search and negative in neuropathology, also the CNV profiling proved that those are tumor cells. I think this is very important because, I mean, showing that in those 3 samples, the classical approach did not detect anything. We not even detected cells, but we surely detected tumor cells.
Thank you very much. There is another question from Alison Whitby. I'm not sure. Do you have the microphone?
Yeah.
Okay.
So I was just wondering what you think about other types of molecules that you can detect in the CSF. So particularly I'm interested in looking at metabolites in the CSF. But whether you think that they would be reflective of the actual tumor or not. You might not have looked into this area, so it doesn't matter if you don't have an opinion. But yeah, I was just wondering if you had any opinions on whether metabolites in the CSF are reflective of the tumor, just like you think that circulating tumor DNA in the CSF is very specific for the tumor.
So as you, I think, quoted correctly, we did not do that. So we are not experts. I mean, of course, we looked into this. I mean, we have not done not a single metabolite analysis with CSF, but of course it's interesting. I think it's a much more direct and easy approach. So if you identify metabolites for a certain setting, sure, I think this is very important and actually could also complement the cell side very much, because again, metabolites, they are free from the cells, you can use the supernatant. I mean, the important step, I think, is this is valuable material. So if we set up studies, we should always think about getting most out of it. And I mean, this is also what we try to do here, trying to analyze as many different analytes from this very precious limited sample. And I think these are strategies you should go for. And if you're interested, just contact us. Doing a study on looking for the tumor cells and on the metabolites could be very interesting. We didn't do that.
Yeah.
Thank you. And I don't know, Claire, if we have a room for another question. Gianna Di Sario.
I think we have one question left, so I'm happy to go through that last question.
Hear me?
Yes.
First of all, great talks. Thank you very much. I was interested in multi-omic approaches. I don't know, because you mentioned them, Bernhard. I don't know if you ever tried anything like that. And in particular, I was interested in the GNT protocol. So thank you in advance for your answer.
Yes, we have done that. So maybe you can mute your thanks. Yes, we have done that. Not in this classical approach. Now, when most of you maybe think on this GNT approach, this is very streamlined protocol. So what we did is our in-house method. As I showed, we get the single cell. We basically divide DNA and RNA, amplify both of them, and we did that in this rare brain cancer, pediatric brain cancer code, where we did from the same single cell RNA and DNA analysis. And I mean, we couldn't show this data today. This is also published soon. I mean, hopefully, really in the next weeks, because we're just finalizing the proof of concept here. So because those were cancers where we didn't have any marker. So basically selected negative cells and looked first in the gene expression, could show that the negative cells are neurological cells, and then proved by the DNA out of the same single cell that most of them, nearly all of them, are tumor cells. So we use this approach in our in-house method, and this is an upcoming paper. And we are also working on single cell genome and proteome sequencing by basically introducing the proteomic workflow in the sequencing workflow. But this is still tech work to do here.
Thank you very much.
Great. Thank you so much to Bernhard, Cäcilia, and Niccolò for giving your time today. It's great that we were able to answer all of the questions. I'm sorry to anyone who was in a rush that we've run over time a little bit. And I'd like to thank the whole audience for joining us today. We will send a short survey around in the next few days, and we'd really appreciate any feedback that will help us improve our future events. It's also useful to get some ideas from you about what topics and themes you would like in these sessions. We plan at the EACR, we plan monthly webinar sessions, some public and some exclusively available to EACR members. You can check the events page on the EACR website, eacr.org, for all of our upcoming events. And this includes all of our conferences and our annual Cancer Research Congress, which this June is in Torino, Italy. I just want to say thank you again to Cäcilia, Bernhard, and Niccolò. Is there anything any of you would like to say before we end the session?
Just want to say it was a pleasure, and thank you, Claire. Thank you, Niccolò, and Cäcilia. Great talk. Thanks.
Thank you.
Thanks to all of you, and I'm very happy to present everything now.
Great. So thank you again to everyone for joining, and we hope to see you again very soon. Enjoy the rest of your week. Goodbye.
Bye.
Bye.